All right. Good afternoon, everybody. Welcome to the November 17th edition of the virtual symposium. Today we have a wonderful session with three professors. I'm trying to get the slides up. Sorry. Where are they? Here we are. For the share screen. Sorry. Oh. Yeah, sorry. Yeah, today's talks we have Professor LaLonde from Reed College, Professor Miles Johnson from the University of Richmond, and Professor Jeff Johnson from Hope College in Holland, Michigan. So today's sessions, you might have noticed, are all focused on research at undergraduate institutions. And I'm really excited to bring this to you because personally, I'm a product of a PUI, having done undergraduate research at Bucknell University before joining industry, and what I learned there has served me really well. And I think the complexity of research that's been going on in these labs is really going to astound you. And I'm really excited to promote research that's going on here, as well as other PUIs around the country. Rachel, if you want to switch to the next slide in the sequence. I can't share mine because you've got, there we go. All right, I'm going to hand it off right now to Steve. He's going to talk a little bit about our upcoming ACS meetings.
All right, morning, afternoon, everyone. So I'll just say a couple of quick words about the San Diego meetings and some related topics. So we're, pretty hard at work planning for the San Diego meeting, which is billed as a hybrid meeting right now. So first I'll say that we have, in the Division of Organic Chemistry, 950 accepted abstracts, which is phenomenal. For context, it's about 50% higher than any of the recent pre-COVID meetings. So it should be a pretty busy meeting. Over 90% of folks indicated preference for an in-person presentation, which we have been able to accommodate. So the meeting will be mostly in person. As you can see, there's going to be about 57 in-person or hybrid oral sessions, about 6 virtual oral sessions, and a lot of posters. So there are a ton of award and organized symposia. This will all be on the DOC website and social media. But if anybody has any questions, please direct them to me. If you could move on to the next slide. I do, want to just quickly point out, the awards that have been the national awards and some of the Division of Organic Chemistry awards, all of which will be presented in the division, kind of roughly spread out through the 2 meetings in 2022. The Corey Award went to Abby Doyle, the Brown Award to Ben List, who also obviously has won the Nobel Prize this year. The Gunther Award went to 3 people over the last 3 years: Sarah O'Connor, Bradley Moore, Ted Polinsky. The Earl Barnes Award to Kevin Campos. The Breslow Award to Reza Gadiri and John Sessler. The Summerjai Award to Jin Huan Yu. The Cope Award to John Hartwig and a number of scholars. And we'll also be we'll also have the industrial early and mid-career investigators, as well as the technical achievements in the spring, and a number of awards that are listed in the fall. Next slide. So I do want to put a quick plug in for the industrial early and mid-career investigators. First, congratulations to all of the 2021 awardees. The nominations for both the early and mid-career awards are currently open. The co-chairs are Lisa McRae and myself. The deadline is February 11th of next year. So please submit a colleague. I'll hand it back over to Scott.
Excellent. Thanks, Steve. And congratulations on your choice as award winner there. So because we've actually missed the last couple of years of last year's of meetings, we also are having our normal fall technical award ceremony for 2021 at the spring meeting. You see the awardees for 2021 listed there on the right. These are awards to recognize BS/MS chemists in industry and government service for their contributions. So the applications for the 2022 awards, which will be shared later on in 2022 at the fall meeting, are open until January 21st, 2022. So if you know some folks that would be good for this award, please go ahead and nominate them by mid-January. Thank you very much. Rachel, if you go to the next one, please. And what was the NOS 2021 is now 2022 at UCSD. I'd like to welcome folks who are going to speak there, the folks listed here on the slide, and anybody who will be joining up and attending that meeting this is in June at UCSD. So I think registration should open up pretty soon next quarter. And then next slide, if you could, please, Rachel. And because we're off a year, we'll have an NOS in 2023, and that will be run by my colleague Michelle Tran-Dubé and our own Steve Silverman here. And that's next year from next summer. And if you know some places that fit the criteria of 1,000 or so people in a lecture hall and a bunch of posters space and such, please give Steve and Michelle a shout out, and they'll have a look at all the nominees for locations before the final decision is made. So that's it for announcements. Thank you very much. One more slide, I think, just to say thank you from Steve, Mark, and I. Sorry. A few list of some next speakers for the virtual symposium for next year: Christina Novato, Nicholas Ball, Hans Renata, Mark Levin, and Mark Ondari. Have been all tentatively signed up for next year in the first half of the year. So we're really excited to keep that going, and looking forward to some excellent discussions about high quality chemistry for next year, as well as keep it going from this year. So thank you from Carl and Steve and myself for all your attendance and joining with us this year. This is our last session for 2021. And again, really excited to have it be focused on undergraduate research at PUIs around the country. We have 3 great speakers with a wide array of different chemistries being talked about today, and we really like to raise awareness of these folks who are doing a huge job teaching the next generation of chemists, doing research, mentoring, coaching, all of the things that they have to do every day to be the best teachers, to give the students the best foundations. Just so proud of all of them for everything they do, and all the other PUI professors out there who are doing a great job in supporting their students and putting out great research. So this is for all of you folks. Thank you very much for all you do. I got my start there, and so did so many other people, and really excited to highlight that today. So our first speaker is Professor Rebecca LaLonde from Reed College in Portland, Oregon. She actually graduated from Reed as her BS, and after a master's in Stanford, earned her PhD from Dean Tost at UC Berkeley. After stops at Dow and Genentech, where she was contributed to the discovery of Arivedge, which is a treatment for basal cell carcinoma, Rebecca returned to Reed in the fall of 2013, where she's now associate professor. She's an avid outdoors person and paragliding pilot, which is really cool. She's really concerned with the environmental impact of organic chemistry, and her research interests are involved in developing non-toxic catalysts derived from bismuth. And these catalysts will be used to synthesize new bonds that are critical for drug and agrochemical research. And she teaches introductory organic chemistry and advanced synthetic organic chemistry, and we'll be talking about the challenges and the opportunities with 2 case studies from generating publishable results from undergraduate researchers. So Rebecca, thank you so much, and take it away.
Thanks so much, Scott, for the invitation. I'm really excited to share what my group of undergraduate students have been able to accomplish over the past 9 years. So for those of you who are unfamiliar with Reed College, We are a private residential college in Southeast Portland, Oregon, and we're really a research-focused institution. So we're a small institution of around 1,400 full-time students. We have about 20 to 25 chemistry and biochemistry majors every year. And our whole educational program is really driven by a capstone course that every single one of our students has to complete. That is what we call a senior thesis. And so this is a year-long independent research project. And to kind of highlight how seriously we take this project, the students actually must pass a junior qualifying exam in order to advance to their senior year and their senior thesis. And so everything that we do at Reed is really focused on research with undergraduates. And I think that it's really critical to highlight how we have to balance different factors within this paradigm. So of course, we still have to have impactful work. And at the same time, we have to have work that is of educational value, right? That is connected to their coursework so that they can see how their coursework impacts research, and of course, preparing them for their work after graduation. It also has to be achievable, right? We're working with untrained, unskilled undergraduates who are just learning, so we have to hold safety as paramount. It also has to be work that can be done in discrete blocks of time. So some of our students work 2 jobs. They only have a couple hours per week to come into the lab to do research. They have to be able to make meaningful progress in that time in a semester or 2 or a summer. We also have to highlight quality. We need to be able to reproduce our results, right? One student might be able to achieve a result and then the next student might have trouble. So we have to really hold the quality of our work high in mind. And also, of course, budget. We don't have incredibly large budgets that can fund huge projects like, for instance, R1 institutions might. So to highlight these issues, I've chosen 2 case studies. And the first is more of a traditional story in medicinal chemistry that brings me back all the way to 2013 in my first group of research students. So this is Rachel Yan, a member of the self-proclaimed Lady Lab. And Rachel came to me in late August and told me that she really, really wanted to do her thesis work in studying breast cancer treatments. She was personally impacted by breast cancer with a family member who had suffered greatly from the disease and was really incredibly motivated to study breast cancer treatments. And to be honest, I was really skeptical at first, right? I've worked in industry, I've worked on these large teams in the pharmaceutical and agrochemical industry and really witnessed firsthand the amount of effort that it takes to put into a drug discovery project. And so I told Rachel that I'd be willing to help her, but that we would really have to come up with a very specific structural hypothesis to test. And so Rachel went off into the literature and started investigating the history of breast cancer treatments, starting with this molecule, tamoxifen. So tamoxifen was actually discovered originally in 1966 and is still used today in the treatment of breast cancer. Unfortunately, it and its bioactive metabolites such as 4-hydroxytamoxifen has partial agonist activity in uterine tissue, which is correlated with an increased incidence of uterine cancer. And so now we have 2nd and 3rd generation selective estrogen receptor modulators. Raloxifene is an example of a 2nd generation SERM. And this is an antagonist in uterine tissue, but unfortunately has poor bioavailability and is utilized now as an osteoporosis treatment instead of a breast cancer treatment. And so all of this led us to a question: can we remove or reduce the agonist activity in uterine tissue of the tamoxifen scaffold while still maintaining the good bioavailability? And so in order to design this better serum, we dug into the structure-activity relationships. And so the SAR of these molecules really show how the sidearm motif is critical. And so it turns out that there's an exposed negative charge on helix 12 in aspartate 351, and that this is necessary for co-activator binding. And so raloxifene has a strong and short hydrogen bond contact, 2.7 angstroms, whereas tamoxifen has a sidearm contact that is much longer, over an angstrom longer. And so this led us to our hypothesis that if we actually could just switch the sidearm position on the tamoxifen scaffold to the meta position, that we might actually be able to better align it in the binding pocket with the raloxifene position. And this would create a shorter and stronger hydrogen bond. And that would hopefully also maintain the bioavailability of the tamoxifen scaffold. And so, just a quick energy minimization and overlay showed that that hypothesis would be correct. And then we moved on to actually downloading the co-crystal structures of 4-hydroxytamoxifen as well as raloxifene in the estrogen receptor alpha binding site. And then we removed the known compounds and docked and minimized our analog and showed that actually this should generate a 2.7 angstrom binding contact. So with that great hypothesis in mind, we moved on to trying to figure out how to make these molecules. So historically, these tamoxifen analogs are made by a McMurray coupling. And you can also put a meta substitution with a PIV protecting group to make 3-hydroxytamoxifen, which is called droloxifene. But if you'll notice, this actually has the wrong stereochemical relationship. Our analog, we need to have the meta substitution proximal to the 4-hydroxy group instead of to the phenyl group. And so Rachel found a 3-component coupling that was used to make droloxifene that is in a non-diastereoselective manner and started to actually try to make our analogs. She was actually really, really successful, but this was where I encountered my first hiccup in working with undergraduates. Rachel graduated after completing her senior thesis and handed this project off to the next student. And the next student was unable to reproduce the results. That student ended up deconstructing this 3-component coupling into an allylation and then arylation. And then the next student wasn't able to reproduce their results. And so what I eventually figured out was having students work in pairs is critical so that you have built-in error bars and that students can immediately verify each other's results. And so I'm going to spare you a lot of gory details, but instead fast forward to 2019, 5.5 years later, when I had 2 extremely talented students who had managed to scale up 15 grams of this intermediate and were busily transforming it by migrating the double bond and deprotecting the PIV group in our microwave. And we had generated 2.5 grams of these separable diastereomers. And so all we had left to do was to alkylate and then deprotect. And then we got this unexpected crystal structure. And so if you look carefully at this crystal structure, you'll realize that it's actually this molecule, where somehow, somewhere along the line in our arylation reaction, we ended up isomerizing the molecule. And so instead of the desired compound, we had this unusable isomer. And I have to confess that fall, I might have shed a tear or two about this, but it actually led us to a new stereoselective method that was based on a method published in 2017, which was a diastereoselective tosylation followed by a palladium catalyzed cross coupling, then simple TBAF deprotection followed by alkylation with 8 different side chains of varying lengths and substitution, eventually led us to a series of 24 analogs with a 4-hydroxy, a hydrogen, or a methyl ether. And the 2 students who were working on this were just amazing. So they not only synthesized all of these 24 analogs, completed the whole characterization of all of the new analogs, but they also measured in triplicate the binding affinity for not only estrogen receptor alpha, but also beta. And what we found was really exciting, that the 4-hydroxy series had 2 compounds, so a pyrrolidine substitution with a propyl linker, as well as the dimethyl substitution, again with the propyl linker, that maintained similar binding activity to tamoxifen. And so it's really unbelievable that these students were able to do this in an incredible short amount of time. So we found out that we had the wrong compound in October. And as you all know, in spring 2020, the labs shut down. They had actually already finished this from October until about mid-February, which is just unbelievable. Both of those students are in graduate school And I really look forward to what they do in the future. And so now I really want to switch gears to a completely different topic, and that is the formation of secondary organic aerosol. So this is an environmental chemistry project, really. And so those of you who are unfamiliar, secondary organic aerosol is a primary component of PM 2.5, which is a toxic component of smog. And so the way this is formed is that there are biogenic volatile organic emissions. So methane is a large portion of that. There are also isoprene and terpenes that are emitted by plants. Those undergo a variety of oxidation processes by UV light, OH radicals, and ozone, and then also by NOx species that are emitted from human pollution sources. And so oxidation of these terpenes by NOx is actually accounted approximately for 50% of the secondary organic aerosol generated in the United States. And so of this is really generated by a lot of the biogenic VOCs that are not from methane. And so things like isoprene, limonene, carine, alpha-pinene, and beta-pinene. And so why is this even important to organic chemists? So studying the reactivity of these oxidized species within secondary organic aerosol is actually critical to our understanding of the role of NOx in our environment and really understanding climate change. And so from an organic chemistry perspective, there's actually a multitude of opportunities. So from our perspective, there's just a double bond that gets oxidized to form compounds like these, alpha hydroxynitrate esters. These are then implicated in the formation of secondary organic aerosol. So the composition of these secondary organic aerosols are largely studied by mass spectrometry. So environmental scientists will go into the field, they will gather environmental samples or generate samples in chamber studies. And what you'll notice is that from this pathway of beta-pinene is that there are a multitude of possible products, and many of them are isomers. And so it's really difficult, if not impossible, to obtain these detailed structural information of complicated mixtures, and also difficult to study the reactivity differences between these isomers using things like mass spec. And so the preparation and the structural characterization of these types of alpha hydroxynitrate esters of terpenes was just not documented in the literature. And in fact, some atmospheric chemists were calling for this so that they could actually create NMR spectral libraries so that NMR and other more detailed structural tools could be used in atmospheric science. And so we went about creating a method to actually make and characterize these alpha hydroxynitrate esters from terpenes like alpha-pinene. We discovered things such as this, whereas atmospheric chemists really had proposed these two primary nitrates. We don't think that these are actually prevalent because the carbocation intermediate actually rearranges quite quickly to generate these types of nitrates instead. And we were actually unable to ever observe or isolate these and instead found different compounds. And this was work published in an atmospheric journal last year. And so now we actually are able to move on to studying the reactivity of these compounds. And there are a lot of different ways that these molecules can react, right? You can photolyze them, you can hydrolyze them, they could undergo sulfolysis reaction or further oxidation, or even heat degradation. And unfortunately, because these molecules hadn't been synthesized previously, the reactivity really had been studied on these sorts of simplified systems and model compounds. So taking a step back again, and this is classic, intro-organic chemistry. From an atmospheric chemistry perspective, these compounds were proposed to mainly undergo hydrolysis from a carbocation to diol diastereomers. But of course, as organic chemists, we know that an epoxide could reform. There could be elimination pathways to form even nominally oxidized species. And so when we actually started studying these reaction mixtures, what we found is that depending on the protic content, there was a differing array of products. That is, the relative humidity of a mixture of aerosol is actually going to impact the product distribution. And in particular, we found this unexpected rearrangement product that was dominant in 100% water solutions. This unexpected rearrangement product might not be interesting from an organic chemistry perspective, but it actually has a very different proposed and predicted vapor pressure. And because this has such a different vapor pressure from the proposed dials, that has implications for how this new dial can then continue to react in the atmosphere. This system is also really interesting because it allows us to study the kinetics by NMR. And so this is a plot of the reaction over time. So here's a signal corresponding to our starting material, as well as one of our dials. You can note that there's an induction period observed in the formation of this major cis dial. And that induction period is correlated with an epoxide formation that actually happens early in the reaction. Furthermore, the consumption of this nitrate appears to be first order. That's not unexpected. And we also found that the major and minor diastereomers are actually only accounting for about 60% of the mass balance of the material. And so other reaction pathways that were not previously considered by atmospheric chemists actually account for 40% of the mass balance, which is a large portion. And so this is really an area that's rich with opportunities that are extremely educational. It's really directly correlated to the intro-organic curriculum and is simultaneously impactful to a field outside of organic chemistry. And so we just submitted this work to Atmosphere as a special issue that actually highlights the interface between synthesis and atmospheric chemistry. And we're also submitting our kinetic studies hopefully soon. And so just a quick summary of some of the lessons that we've learned from these unexpected adventures. And first is, you're taking a step outside of these traditional areas of interest can actually be incredibly fruitful, particularly if you find a system of study that can lead to multiple avenues of investigation and also is really directly connected to coursework. In addition, what I found is, teamwork is actually essential in undergraduate work. So it's critical for safety. We have built-in safety with our lab spotters, right? The students are always working in pairs. It helps us verify the reproducibility, also assists in time management. One student can set up a reaction and another can monitor it or work it up. And it's also invaluable practice for a collaborative workplace. The students have to be able to communicate their results and work effectively together if they're to, for instance, do a reaction in tandem. So finally, I need to thank all of the students that actually did this work in the lab. This was my lab group this summer, which is an incredible group of students to work with. I also need to thank the Research Corporation for funding, as well as Reed College for startup funding. My main collaborator, who's an atmospheric chemist, is Anthony Carasquillo at Williams College. And it's just been a really phenomenal collaboration to be able to work in tandem with an atmospheric chemist on this atmospheric chemistry work. And as always, Izzy, the lab mascot, says safety first, and I'd be happy to answer any questions.
All right, awesome. Thank you so much, Rebecca. I was just, while we're waiting for some questions to roll in, the analysis of these, I mean, these are, what kind of instrumentation are you teaching? Are you having the kids, the students look at GC and NMR and things like that? How does that run in your undergraduate lab?
Yeah, so Reed is actually really fortunate. We have a huge array of instrumentation. So we have a 400 megahertz NMR that we have an auto sampler for. So if we need routine data, we can just, the students go down themselves and submit their work. And it's also, we use that for our 2D, pseudo 2D NMR kinetics. We've also used, we have a GC mass spec, but unfortunately a lot of these compounds actually isomerize and react when they're volatilized. And so we don't, we tend not to use GC mass spec as much. We also use LC. We've tried using React IR also for these measurements. And so the students have really gotten familiar with that. It's really an amazing area where the students get exposed to a variety of instrumentation. for sure.
Okay, so there's there's a question in the chat here about asking you to comment on the inability of one student to repeat the research of another student. I mean, to me, I think that's probably somewhat common in undergraduate, but maybe you can speak to that.
Yeah, I mean, I think it is common in undergraduate research because I can't stand over a student and watch exactly what they're doing all the time. And because they're really at the very beginning of their training, you never really know exactly what they've done. And sometimes it's a delicate procedure that one student happens to be talented and is able to achieve the result. And then the next student might need a little bit more learning and growth to get to that point where they're able to reproduce a result. But I also just want to point out, reproducibility has been pointed out as potentially problematic in the chemistry literature, right? It's not just undergraduates. I think a lot of us have discovered papers where we go and try to reproduce a published result and can't.
Okay. And here's another question that popped up in the Q&A. Can you talk a bit more about getting the samples for atmospheric chemistry? I think you said you went to the field and got them. What are the challenges with undergraduate students doing this?
Yes, so I am not a field scientist, but I collaborate with field scientists. And so one of the things that atmospheric chemists do is they actually put together large grants of literally dozens of folks that then go into the field and have a summer field campaign where they set up collection areas and they generate these filters that have collected particulate matter. Those filters are then brought back to the lab, extracted, and then analyzed. And so one of the things that I didn't talk about, but that we're working on is actually using these synthesized standards to now identify components using NMR. And so that is challenging because it's very small quantities. And so we're working on getting access to instruments with much higher signal to noise, such as a cryoprobe, as well as working with other types of NMR experiments, such as DOSY NMR, to be able to separate out components in these mixtures.
Excellent. Okay, and I'm going to go one last question before we switch speakers, and this is from Scott Seaberth, from professor from Temple. Can you? I said lovely. He says lovely work. Can you comment on isomerization of where the meta and para arrow groups seem to switch places? I believe that's in the beginning.
Yes, that's still honestly a mystery that I have not gone back to investigate because I really just wanted to get the analogs made and to get the assay data taken. But the yields for that reaction were pretty low. So the aerolation, we optimized finally to 50% yield. And there were a couple of rearrangement products that we found in that mixture, but we never really went back to investigate fully what they were. So unfortunately, I can't really totally tell you the answer to that.
All right. Well, that was a wonderful presentation. Thank you so much. Amazing work going on in your lab. Thank you again for coming here and sharing your stories. Really appreciate the effort your students are putting in, the effort you're putting in to teach them. It's fantastic. Thank you so much for being part of this symposium today. All right. So if there's any, and if I think if there are other quick Q&As, Rebecca can answer them in the chat in the Q&A window afterwards. So we're going to move on to Miles right now. So our next speaker is Professor Miles Johnson from the University of Richmond in Richmond, Virginia. Interestingly, and I did not do this on purpose, but Miles also earned his PhD from the TOS lab at Berkeley. And that was completely didn't mean to do that, but that's all right. I'm glad to have Miles here because he and I collaborated on the gold catalysis project back when he was doing his PhD at Berkeley, and I still have remnants of that here around my lab, and it was an enjoyable time. So thank you, Miles, for that. And but after Berkeley and postdoc with Professor Jonas Peters at Caltech, Miles moved to Richmond, where he is now assistant professor. And the Johnson Lab focuses on the design of new transition metal catalysts able to convert single chemical feedstocks into fine chemicals, as well as design new ligands and synthesis of organometallic compounds. So I'm really excited to have Miles here and to hear him talk about what's going on since the last time he and I spoke a few years back. So take it away, Miles. I think you're still on mute, Miles.
Sorry, my laser pointer froze up on me.
That's all right. Thank you.
Okay. All right. So thank you, Scott, for the invitation today. And thank you all for coming and participating in this great opportunity. Today, I'm going to tell you a little about the work that my lab has done over the past 5 years concerning the synthesis and study of aminophosphine ligands, in particular with a focus on bisphosphinoprol ligands. And before I dive into the science, I just want to highlight the students who have done this work. So I've had the opportunity to mentor 18 amazing students since 2016. And the ones who did the majority of the work in this presentation are Hilary Fuqua, Isaiah Duplessis, Sophie Weinberg, Jess Snelling, Sadie Taylor, and Julia Vidlak, and they'll be highlighted throughout this presentation. So our lab is broadly interested in the development of new ligands and taking these ligands on to explore their coordination chemistry and see what impact they have on the metals to which they're bound. And then from there, once we've established that fundamental coordination chemistry, we go on to look at the fundamental reactivity with the end goal of developing catalytic reactions that solve problems such as sustainability, selectivity, common things that as synthetic chemists, we're still trying to sort out with any given reaction. And within the context of developing new ligands, we've chosen the motif of aminophosphines. And there are two particular classes of aminophosphines that we've chosen to study, proisophosphotranes and bisphosphinoprols. And a unifying characteristic of these two is the modular synthesis of both types of ligands, which is essential in an undergraduate lab, having ligand framework that is not 10 linear steps, but rather can be separated, can be prepared from a single common intermediate to potentially make libraries. Also, we're interested in conformational flexibility, which is a key component of proisophosphotranes. And when I started at Richmond, I wanted to look at both of these classes of ligands, but proisophosphotranes had already been established as ligands. And so we're a good point of departure for us to first focus on this established ligand that we could buy and just have from a commercial source, while also developing the chemistry of bisphosphinoprols, which were completely unexplored in terms of reactivity. And so before diving into the phosphinoprol chemistry, I just want to highlight our initial work studying proisophosphotranes, which in part sets the tone for how we approach our chemistry. So Adrian Matthew and Greg Gravalis did tremendous work making palladium proisophosphotrane complexes to study a long-standing hypothesis that proisophosphotranes can undergo conformational changes during catalysis. And although there were many studies by Vercade and coworkers on palladium proisophosphotrane complexes generated in situ, there had never been discrete palladium proisophosphotrane complexes that had been studied in terms of their elementary reactivity of elementary steps, as well as to examine the hypothesis of conformational changes. And so these two students picked apart a catalytic cycle, examining each step, isolating the intermediates, growing crystals, and examining both stoichiometric and catalytic activity. And they were able, Greg and Adrian were able to show that indeed there are conformational changes. So these are the sort of questions we're looking at. And for more on this, please see this paper in Organometallics and then a follow-up study where we looked even deeper into what affects conformational changes in aminophosphines. And this is work that is still going on, but I think establishes our process of taking a catalytic process, breaking it down, also studying in great depth the ligands themselves. And as we were working on this, we were starting to build up ideas for new ligands. And the first thing we considered was phosphinoperols. So there are numerous examples of phosphinoperols in the literature, and I just want to give you a little bit of background. So two seminal works are those of Allen and Malloy. Allen showed the first example of a 2-phosphinoperol that will figure prominently throughout this talk. And then Malloy showed that having N-phosphinoperols renders the phosphorus a weaker donor than, say, triphenylphosphine, in part due to the electron withdrawing nature of the nitrogen of the perol. And then many of us have seen the work of Beller and Dyer, so these phosphinoperol ligands that are widely used, especially in catalysis in the case of Beller's ligands. So there is precedent for using phosphinoperols in studying catalytic reactions. The niche that we saw or the gap in the literature that we saw was having bidentate bisphosphinoperols. So Semmelhack had shown this ligand, which looks a lot like this ligand from the Allen group, but now with a phosphino group affixed at the end position. And then also this bidentate phosphine, the reactivity for which and for its palladium compound were not explored. And so we decided to go down the path of exploring bisphosphinoperol ligands similar to these ones. And we tried to think about what would the niche be that we were filling. And as probably most of us have seen, bidentate bisphosphines are everywhere, and they are essential for advancing our field of synthetic chemistry. They have exceptional performance. They've been highly tuned to work well in a variety of applications. Many of them are commercially available. They are wonderful go-tos to solving many of our synthetic challenges. However, there are some limitations. For example, some of the syntheses of these molecules can be lengthy, require multiple steps, multiple purifications by column chromatography. Although these are commercially available, often they're expensive. In the case of the bisphosphinobenzene motif, you have the potential for unwanted benzyne formation and establishing that installing the first phosphino group. And also, these ligands are often tailored for palladium. And my group has the goal of producing more sustainable catalytic processes. And so we want to try to develop ligands that might be specifically advantageous for, say, nickel and copper. And also, even in the synthesis of some of these ligands, palladium catalysis is needed for the CP bond formation. And so we're trying to find ways to synthesize new ligands without depending further upstream on methods that might be costly or environmentally detrimental. And so this led us to thinking about this phosphinoperol group that was originally synthesized by Allen, and the synthesis for which was refined by the Tonks group back in 2014. And Tonks had also shown that you could easily depronate this phosphinoperol to make the lithium pyrrolide. And so we wondered if we could take this compound and then treat it with a variety of chlorophosphines to make bisphosphinoperol ligands. And even just glancing at SciFinder, there are dozens of chlorophosphines that are commercially available, allowing the potential for large libraries to be formed. So we envisioned a modular and simple synthesis that uses a stable intermediate. We can synthesize multiple grams and have it stored at room temperature in a glove box indefinitely. And also these ligands may impart unique electronic properties that could be beneficial to the sort of catalysis that we would like to pursue. For one thing, with this nitrogen installed, could attenuate the donor properties of this phosphine and perhaps encourage reductive elimination and reactions for which reductive elimination is the rate determining step. And also this unsymmetrical molecule could also favor reductive elimination, as has been seen in a couple of studies where unsymmetrical phosphines were found to bring down the barrier to CN and CC bond formation. So we started out with Hilary Fuqua, who began this project, established the first synthesis of the isopropyl variant of our ligand, and Sophie Weinberg, who worked with Hilary to generate this library. And they found that under mild conditions, just using ether, room temperature for 4 hours to overnight, they could synthesize 10 different phosphine ligands. All of these in a single step from a common intermediate, and all of them can be purified by simple filtration These yields are actually after recrystallization, just for thoroughness, but filtration is sufficient. This method tolerates aryl groups with varying electronic properties, whether it's the electron donating para-anisile or electron withdrawing para-CF3 substituent. Also, mesothelin ligands are tolerated. We also were able to prepare the diisopropyl and dicyclohexyl variants and the terpbutyl phenyl, which the diterpbutyl case does not work, but the monoterpbutyl phenyl case does, just as a side note. We can also make phosphoramidites, which have been found to be privileged ligands, particularly in enantioselective chemistry. And given the electron withdrawing nature of these phosphoramidites, we could expect that they might be beneficial as well for reactions in which having a more electron deficient metal is beneficial. So with these ligands, and I always joke with my students that it's great you've made a phosphine, but until we have it on the metal, it's not quite a ligand. But Hillary, Isaiah, and Sophie took our prototypical diisopropyl ligand and were able to metalate it with a variety of late metals. So they first treated it with platinum chloride to make this square planar platinum dichloride. Similarly, they're able to make the palladium chloride in high yield. They're able to form a dimeric copper chloride complex. And finally, a nickel dichloride complex. And we were very excited about these results because we want to make ligands that could be broadly applicable to a number of metals, and in particular, be compatible with first row metals. And as seen with the copper and nickel, this goal was achieved. And I'd like to highlight at this point that all the crystallographic data collected for the past 5 years was conducted by our amazing collaborator, Professor Nathan Schley at Vanderbilt. So now that we have these complexes, we want to look in depth at how the pyrrole linker affects the steric and electronic properties of a given compound or of a given complex. And so Julia set out to make the nickel dichloride complex again, and also making a nickel dicarbonyl compound. And simultaneously, she made this known phenylene analog. So again, treating diamine nickel dichloride with the ligand to get the corresponding nickel dichloride complex and the crystal structure. She was able to grow crystals to obtain the structure. And also treatment with the bis triphenylphosphine nickel dicarbonyl, she was able to get to this nickel dicarbonyl compound. So with these 4 compounds in hand, she was then able to pick apart the steric and electronic contributions for each of these ligands. And she began just looking at classic CO stretches. And she found that, consistent with what's been found in the literature and with our expectations, this phosphenopyrrole ligand has attenuated donor ability, so rendering the nickel less electron rich, and as a result, having a stronger CO bond, and as indicated by these higher energies in the red trace. Whereas with the phenylene analog, those values are shifted to the right due to the more electron donating nature of the benzene ring. She then wanted to examine the electron donating ability of these ligands by electrochemical studies. And so scanning cathodically, she was able to find that, again, consistent with our expectations, that the nickel center would be less electron rich in the case of the phosphenopyrrole complex. She was able to reduce the compound at negative 1.6 volts, whereas with the phenylene compound, the reduction took place at negative 1.7. So again, consistent with the idea that this nickel center is richer than this one. So having established the electronic properties of these ligands, Julia then set out to look at the steric ones. So first, she took the crystal structures and just measured the bite angle. So as a proxy of steric bulk, she found that these vary by only about 1%. Or by 1 degree, so a small deviation. And wanting to delve even further, we looked at the buried volume. So to get an idea of what percentage of a sphere that encapsulates the nickel center, how much of that is occupied by the ligand. And again, we have variation by only 1% or by less than 1%. And so taken together, it seems that the steric properties of these ligands are similar, but electron differences remain. And so here's just a quick summary. So again, electronically, the pyrrole complex is electron deficient, and the pyrrole ligand is less electron donating to the nickel center. Electronic differences do not transfer to the HOMO-LUMO gap, which we determined by UV-Vis, so very similar orange complexes. And then looking at the steric parameters, we found the bite angles were similar and percent buried volume. And then there's another subtle detail that we wanted to look at, and that was the trans influence exerted on this nickel-chlorine bond that's trans to the phosphino group that's either bound to nitrogen or bound to carbon. And we found that in the case of the phosphino group bound to nitrogen, this phosphine donor imparts a less strong trans influence as indicated by its shorter bond relative to the diisopropyl phosphino group bound to the benzene ring. And this is important because we now have a motif where we could potentially influence the trans influence of the donor ligand without perturbing the steric properties. And in a couple of studies, it was found that by having this asymmetry and electronic properties, you can promote reductive elimination. Notably, both of those studies were in palladium chemistry, and so the impact with nickel has yet to be explored, though is something we will be studying. So having made these complexes, made these ligands, we then wanted to explore catalysis. And so Julia, Hillary, and Sophie initially took nickel cod in the presence of 4-chlorobenzonitrile and morpholine, and found that they could get some coupling, albeit in relatively low yield. And then we moved on to this primary amine, so furfurylamine with 4-chlorobenzonitrile. And we chose this system because it's so well benchmarked, particularly by the Stratioto group. And we found that we only got about 22% yield. And we thought that this might be due to the thermal instability of nickel cod and some of the detrimental reactions that can arise from having cyclooctadiene present in the reaction. And so Julia decided to make a discrete nickel complex using this TMEDA nickel chloride orthotolyl starting material. So with gentle heating over a few hours, she is able to isolate analytically pure nickel complex that is also air stable. When she subjected this complex to the reaction conditions that we saw previously, she was able to achieve a yield of 71% at room temperature, and with slightly elevated temperatures, full conversion to the product. So with having established that these are competent catalysts, she, Sadie, and Morgan Labadini set out to make a library of these ligands. And so far, we've had fairly good success. Only 2 of our ligands have not worked, and those were the ethylene glycolate complex and the para-CF3. And even this para-CF3, Sadie is shown that under the right conditions, you can get it pure. So we're about 7 for 8 now. But the majority, without any special considerations, can be isolated in relatively high yield, and in all but one case, the paramethyl case, all of these are air stable. And interestingly, they exist in a mixture of isomers in some cases, especially when there's not a big difference in steric bulk. So the para-anisyl, para-toluol, and the mixed terpetal phenyl, and which again could point to the difference in trans influence between the 2 donors as well. So not just steric impact. And to show you what we see, when we run these reactions in J-Young tube, we see fairly rapid consumption of the 2 doublets consistent with the phosphine ligand, and then emergence of both stereoisomers. And we're currently trying to unambiguously characterize which isomer is which in each case. And once we've gone back now with our new library of ligands, of nickel orthotolyl complexes, we found that we can achieve even higher yield. So isopropyl was not where we maxed out. So with going to bulkier ligands, we are able to get higher yields. But now that we're at the point of developing catalytic methods, we want to approach this in a more systematic way and correlate structure and reactivity. So as all my students know, I love the Tolman parameters, but they are not sufficient for bidentate ligands. And so we were able, during the summer of 2020, when experimental chemistry was not possible, Julia was able to undertake a computational study that summer in collaboration with our collaborator, Professor Kelly Donald, also at the University of Richmond, to look at both the electronic, as indicated by the simulated CO stretches, and the steric properties, as indicated by buried volume, of these nickel complexes. And so now, as we go back to doing more in-person research, we have this template for guiding our mechanistic studies and finding the best possible ligands for certain reactions. So in conclusion, we were able to attain our goal of making new ligands in a modular fashion, applying them to the development of developing the coordination chemistry of some late metals, and both experimentally and computationally, we've elucidated the steric and electronic properties of these ligands and have applied that information to studying catalysis. And now we are looking at different methods to functionalize the two positions so we can have a truly modular system where it's not just the N-phosphino group being modified. We're examining elementary steps and also trying to develop methods more, in particular, carbon-nitrogen and carbon-oxygen bond formation. So with that, I want to thank all of the people who made this possible, all 18 students and our collaborators, Professor Nathan Schley and Professor Kelly Donald, also these funding agencies for their support. And I want to thank you for your time, and I'd happily take any questions.
Awesome, Miles. Thank you so much. It's a great project, near and dear to what we do here in pharma for sure. You know, anything helping us make some better CN bonds is gonna be great. While we wait for some questions to roll in, I just want to say, yeah, that was fantastic. Great design and use. I'm glad you got to try and use your ligands. Any thoughts on other substitution around the carbons of the parole? Would they, whether they donating or accepting or withdrawing on the parole itself to maybe change that phosphine character?
So just modifying the parole linker itself, so it's not just
yeah.
So we've thought about that, but there is the issue of having various stereoisomers. So if you were to have a methyl group at the 3 position, you could end up installing that diphenylphosphino group, maybe in between the nitrogen and the methyl, or maybe on the other side. And then also there's the issue of controlling where that first phosphine is added. Usually we get about a 1-to-1 mixture of 2 and 3 substitution. So it could add a whole other layer of complexity. But that is something we're working on, trying to improve that initial synthesis. And so far we've even improved the column conditions, so we no longer need halogenated solvents, but are not quite at further functionalization of the parole ligand, but it is something we want to try.
Excellent. So we have a question from a Michael Doyle it says, wow, important work and potentially very useful. What causes the geometrical isomer ratios of the nickel isomers that you have observed?
So that is something we're still trying to study. It does not look like they interconvert. And even with trying the reaction at low temperature versus high temperature initially, we tend for a couple of model systems to get the same distribution. So we can't say for sure yet what the cause is, but we plan to do further kinetic studies, variable temperature experiments, just to see if there is an interconversion that we haven't seen yet. But as indicated by the para-substituted phenyl rings, it does seem like electronics play a major role, since you can, I believe, even with the paratoluol, get about a 1-to-2 mixture just by having those two methyl groups act as donors. So we can't, and there's certainly a steric aspect, but even removing it, so more of like a Hammett study on the ligands, it looks like the electrons have an influence.
Excellent. Got time for a couple more quick questions. So one from the YouTube channel from E. Gray says, are the isolated ligand nickel orthotolyl complexes required for catalysis, or can the TMEDA complex be used as a pre-catalyst in the reaction mixture?
We've only tried the reaction once using the generating the catalyst in situ from the TMEDA nickel complex, and we got a much lower yield.
Thank you. And then from Anonymous, we have on the topic of CN formation, are you interested in looking to use these catalysts for potential nitrene transfer, either with cycloadditions or CH transformations?
Ooh, I have not considered that. That's a bit out of our wheelhouse, but I would, once we've established more of the cross coupling, I would like to go on to study other reactions, and nitrene transfer I think would be fun to study with these ligands. So thanks for the suggestion.
Okay, well, thank you, Miles. That's all I see right now, and I think we got to move on to Jeff in the interest of time. But great job. Thank you so much for being part of this symposium today. It's really exciting research, and good luck to you and the students. Thank you very much.
Thank
All
you.
right, so our next presenter is Professor Jeff Johnson from Hope College. And so he's our final speaker today. And after receiving his bachelor's from Gustavus Adolphus University, he earned his PhD from Wisconsin-Madison in 2004, and then was off to Colorado State for a postdoc with Professor Robus. And after that, he moved to Hope, where he's now professor of chemistry, and his research focused on activation and functionalization of carbon-carbon single bonds through understanding of the mechanisms of known transformations and development of new ones. And when reading Jeff's website, I noticed one interesting fact about Jeff is he's an avid and evidently expert curler, and is a 5-time North Dakota state champion, and attended the U.S. Winter Olympic Trials in 1998. And that sounds awesome. So we'll have to hear you talk, and we'll talk some other time about curling, which I've never done. But
all right, well, thanks for that introduction. Yeah, I am still getting traction of that, even though it's about 25 years ago now. So, but with the increasing popularity of curling, I just people keep on bringing it up. All right, well, thanks, Scott, for that introduction and the invitation. It's an honor to be a part of this symposium today and to join Miles and Rebecca. So thank you for that. Thank you for the opportunity. I always like to begin my talks with just an introduction to Hope College. It's not necessarily a household name. So Hope College is located in Holland, Michigan. I get my slides through. There we go. All right. So we're in Holland, Michigan. So it's right along the shores of Lake Michigan on the west side of the state. As the name Holland implies, so the town is the town of windmills and tulips, and especially in the springtime, it's a fantastic place to visit. Hope College is located right downtown Holland. And so it's a private four-year liberal arts institution. We have just over 3,000 students here on campus, 14 tenure track members here in the chemistry department, and we graduate about 40 chemistry majors a year. And the research tradition here at Hope dates back nearly 100 years now, still going strong. And while we do research year round, we have a summer research crew of somewhere on the order of 50 to 60 students are here for a 10 weeks session each summer. And that's when things really, really get pushed forward. All right, so within my group at Hope, I kind of look at two general areas. We like to develop new reactivity, and then we also like to do mechanistic studies to figure out how things work. And so when everything works at its best, these two areas interact with one another. We've developed some new reactivity. Then we go in and look at the mechanistic, do a mechanistic study, figure out how it works, and then that informs how we can develop additional new reactivity. And so these two things can feed each other and really continue over the course of years that I'll kind of describe here in a few minutes. So what I'd really like to focus on today is the aspect of carbon-carbon single bond activation using rhodium. And so here's a couple of examples of some systems that I will work through. So really there's kind of thinking about the idea of CC bond activation. There's two general strategies to make this happen. And so one is ring strain, right? You have a small molecule and you can pop that open. And then the other strategy is the use of enforced proximity. So there's something about a ligand, the substrate, something else that will hold a transition metal close to a reactive site and allow you to actually cleave a carbon-carbon single bond. And so when we're thinking about these methods, there's ongoing challenges that are in many ways very, very similar to things that you see in CH activation. Expanding it to general systems, getting beyond those strained rings, achieving transition metal catalysis in these systems. You can imagine that many of the strategies used for breaking a CH bond also apply for CC bonds and vice versa. And so avoiding CH activation in these systems is a common challenge. And then of course, just to develop general methodology. And so when we started in this area about almost 15 years ago now, the field was small enough that you could actually almost have a comprehensive review of every single transition metal catalyzed CC bond activation reaction. That the field has expanded greatly over the past decade and a half. And so now, just in January, an entire issue of chemical reviews was dedicated solely to this idea of CC bond activation. But when we started, there's really a lack of mechanistic information, and particularly on those non-strained systems. And so that's where we stepped in to this idea to study one of these systems. And so about that time that we were looking for a subject to study, Chris Douglas at the University of Minnesota published this reaction right here. So this is an intramolecular carboacylation reaction in which this carbon-carbon bond right here is cleaved, and ultimately this alkene is inserted in between those two carbons. So this is a fantastic reaction on several levels. So catalytic and rhodium, it's something that actually increases complexity. So that's sort of interest to study. And beyond that, it's a fantastically clean reaction. It is nearly a spot-to-spot conversion. And so that helps make this a great subject of study. And so just an idea of how this reaction actually works, or the proposed mechanism, is we have our rhodium species that coordinates to the Lewis basic nitrogen of the quinoline moiety. Then that undergoes a formal oxidative addition process. So you get a rhodium-3 species here. So it actually breaks into that carbon-carbon single bond. At that point, the alkene can coordinate to the rhodium and then does an alkene insertion in between the rhodium and the aryl bond to generate the species here. And then you get reductive elimination connecting these two carbons, generating that new product, and then also regenerating the rhodium in the process. And so in beginning to look at the study, of course, we have to find a method to do that. We settled on NMR spectroscopy. And so what we would do is run these reactions in resealable NMR tubes, put them in an oil bath at 130 degrees, pull them out, run them down the hallway, and take NMR. And so this worked really, really well. Again, a very, very well kinetically behaved system, but one with an interesting rate loss. So you might notice up here that this actually has a zero order dependence in starting material. One of the fun things in working with undergraduates is they don't really know enough to know that that's not the usual case for an organic reaction, that you have zero order dependence on substrate. But what that tells us about this system is actually this is the resting state of catalysis. And so you can imagine an equilibrium between free rhodium and free quinolynyl ketone in equilibrium with this coordination compound. And essentially this equilibrium lies well towards this rhodium complex. And so as a result, at the initial stages of catalysis, when you have a vast excess of your starting material, this coordinated complex is the only species in solution, and it doesn't matter how much more substrate you add, it's not going to impact the rate. So this is our rate, our resting state of catalysis. And now the question came is, what's the turnover limiting step? What's that highest energy barrier? We had two kind of candidates there. So the oxidative addition, the actual CC bond activation, or the alkene insertion over here. In a typical system, you could like change concentrations and things like that. In an intramolecular system, we're sort of stuck. And so what we did is actually turn to Dan Singleton's method of natural abundance kinetic isotope effects. And so this actually allows us to determine the C12, C13 kinetic isotope effects effectively of all the carbons in the molecule at the same time. And so this method works really well with very, very clean systems and when people have lots of access to NMR time. So this is one of the bonuses of working at a PUI, is that we pretty much monopolized the NMR for a couple of weeks right around a spring break and ran dozens of these reactions that are essentially 20-hour carbon reactions to get the quantitative carbon NMR. So looking at the results of these, what we find is, so using these carbons as internal standards, assuming that they aren't involved in the reaction, we can determine the kinetic isotope effects at each of these positions. So these two alkene carbons here essentially have negligible kinetic isotope effects, so not much action there. But then we look at these two carbons, the carbonyl carbon and the neighboring aryl carbon, those have kinetic isotope effects of 1.026 and 0.027. And as small as those are actually in the range of primary C12, C13 kinetic isotope effects. And so what this tells us then is that turnover limiting step of catalysis is here, that actual breaking of the carbon-carbon single bond. From there, some additional physical organic type studies on this, and we determined the activation parameters. And that activation entropy in particular tells us there's not much rearrangement between the resting state and the turnover limiting step, which is consistent with this mechanistic picture right here. And so the main driver on this initial project here is Colin Rathman. He went on to get his PhD with Jen Prescher at Irvine, and is now an assistant professor at Davidson College in Pennsylvania. Continuing on with the physical organic nature of this, we went on and then made several analogs with different substitution patterns here around this aromatic ring. What we found is different elements that donate electron density into this carbon-carbon bond actually made the reaction go faster. And what that did is it kind of gave us an idea of what actually happens in that CC activation step. So I had always thought about it myself as sort of a concerted activation process. But that's one that actually typically accelerates with electron deficient substrates. So for example, palladium inserting into an aryl iodide bond or something like that. Instead, what this suggests is actually an addition rearrangement type sequence. And so what we think is the rhodium is attacking the carbonyl right here, kicking electrons up. And when those electrons drop back down, we get a Beckmann type rearrangement, something that would go faster with more electron rich substituents. And that's actually the process that cleaves the carbon-carbon single bond to give us this rhodium-3 intermediate. Right, so several students here that helped with that. I'll note Patrick Lutz went on and did his PhD with Abby Doyle out at Princeton and is currently probably interviewing as we speak. And then we have some additional, Tim went off into industry directly. And then between Casey and Bree and John, we have a couple of dentists and someone who's actually doing some missions work now. So an example of how you have students at a PUI who branch off in all sorts of directions once they finish the research. All right, so here, as much as I enjoy those kinetic studies, what I really want to do is develop new organic tools. And so developing these new reactions, we like to combine some of these things that we learned from these mechanistic studies. And so from this initial study that you've seen, we have an effective directing group there, and we have an idea of that oxidative addition process. From another study that I won't be able to chat about, but what we found is that when we have metal aryl species like this, they can be stabilized by orthofluoro substituents. And so what we decided to do is take these two ideas, piece them together, and kind of develop this strategy. The thought is with a molecule like this, we treat this with rhodium, we could get the CC bond activation, get this rhodium-3 intermediate that is now stabilized by that orthofluoro. That might give us a little bit more time for this intermediate that we can intercept it with some sort of a pi system or something like that. And so what we started out is going ahead and trying to intercept this with a series of alkenes and alkynes. And as organic chemists do, like we threw the reagent cabinet at it to see what would happen. And what we eventually found was Michael acceptors, like this methyl acrylate, reacted cleanly with this orthofluorinated quinolynyl ketone. And so the anticipated product here is cleavage of this CC bonded insertion of the alkene to give us this compound here. Turns out that's not what we observed. Instead, what we found was this compound, the alkene. And so what's happening here is presumably we're getting our activation process here. We get the alkene insertion into this rhodium aryl bond, give us this species, but now instead of reductive elimination, what we do is we have competing beta hydride elimination. That happens. This elimination then ultimately gives us this alkene. It does, a couple steps later, regenerates a rhodium catalyst to complete the catalytic cycle. But ultimately we get this Heck type reactivity that's actually really quite clean and high yielding. And so we went from there to kind of establish the reaction scope on this. And I will say that Michael acceptors typically work. Unsubstituted Michael acceptors, mono-substituted species work really well. As soon as we get to 1,2 di-substituted species, the reaction shuts down. That's a hurdle we still haven't been able to get over. It generally works with a number of quinolynyl ketones. So that's essentially this group that's here is donated from the quinolynyl ketone. As you'll note, we get to like kind of neutral species and electron-rich species, the yields drop significantly, but this works really quite well with electron deficient. All right, so overall, we demonstrated with a number of Michael acceptors and different ketones, but that is the big limitation there, that we're limited to mono or 1,1 di-substituted Michael acceptors. And so kind of thinking back to that intermediate, what else can we do with that species? We thought we could also intercept this with an organometallic of some sort, with the idea of if we could get some sort of an exchange process to work, then perhaps we could get some chemistry to happen on this end. And so we were actually able to get this to work with kind of selective choice of starting material. What we can do is start with this specific quinolynyl ketone works the best, this like orthomethoxy species, and we get that activation and then exchange with an aryl boronic acid. And so what ultimately we can achieve in this case is a ketone exchange from one type of ketone to another. Just to give you some examples of this, again, electron deficient aryl boronic acids work really well in this reaction. And so we can get some high yields of this new ketone. And then even neutral species with this, again, the selective choice of our starting material, we can get to any one of these other ketones in just a single step from that. And so this just sort of demonstrates the type of reactivity that we're trying to get to, where you can take one common starting material and get to any one of these quinolynyl ketones through a single step. All right, so these are, again, kind of some notes on that. As electron deficient quinolynyl ketones tend to be the favored product. We can also make some alkyl species, but as soon as you get away from methyl, we start getting competing beta hydride elimination, and so we get some methyl there as well. And so the students on this project, just to note, Joe Dennis did his PhD with Steve Buchwald out at MIT and is now down in the San Diego area. Stana Dorn is at IU with Kevin Brown, and Chad Campagner just finished his pharmacy degree at the University of Michigan recently. So, so anybody who's really interested in quinolynyl species at this point is probably really excited about this chemistry, but the other 99.9% of you out there are wondering what else can you do besides quinolynyl? And so that's really what we're trying to do moving forward here. And so some of our strategies that we're looking at is taking a page from Chol Ho Jan, who has made some imines out of these ketones and then used this group as a directing group to kind of go this approach. We're also thinking about using amides and then kind of, again, appending like a pyridine to insert it into that CC bond. And so we have some efforts going on in that area. What has seen the most movement forward for us, however, is using a pyridyl compound, a pyridine directing group shown here. And so the general idea of this, when our group first came on this, was here is the quinolynyl ketone that we've used quite a bit with that pendant alkene. Well, why don't we simply switch this for a 2-pyridyl group and get the same chemistry? So we treated it under the same conditions, expecting this compound here where we'd get CC activation here and insertion of the alkene. Unfortunately, yet again, we got clean reaction, but absolutely nothing of the product that we expected. This one took a while to dig in and figure out what was going on in this reaction. And it turns out that there are two separate things happening. One that we weren't as excited about is simply a Claisen rearrangement over here. That was kind of a red herring for a while that made us think successful things when it wasn't the case. But the more interesting aspect is right here, we are missing the carbonyl. And so this is actually a decarbonylation process from this pyridyl ketone. And so we were really excited about this point, had all sorts of things drawn up on what we were going to do with this until one of us bothered to check the literature a little bit more closely and realized that she and his coworkers in Beijing had published something very, very, very similar. And so you can see that example here, slightly different rhodium catalyst, a little bit different solvent and so on. The conditions vary slightly, but the same general idea of using that pyridine group, direct the reactivity and get it decarbonylation. There was enough difference in terms of what their substrate scope was and what we've been able to do that we actually did decide to pursue this a little bit more. And so our group made a number of these pyridyl ketones and then have kind of optimized that decarbonylation process. And so we can repeat most of the same substrates that the Xi group did without a problem. Demonstrate a few other functional groups that they didn't publish in their paper, as well as some substitution on this other side that I'll see becomes important here in a few minutes. But then also looked at putting heterocycles onto this. So now we can put this aryl group in the starting material as various heterocycles or even like ferrocenyl compounds and still get this clean decarbonylation process and really high yields. We start seeing some interesting cases of putting some larger heterocycles in there that we get the gearing effect and get some sort of diastereomer type products in there. And we've just recently started exploring getting these sort of congested 1,2,3 tri-aryl benzene species where we can actually control which groups are on either side. Again, going through this acylation process and then this decarbonylation to get there. So again, looking at this, there's clean and high yielding reactions. Most of these are really nice and easy to run. And nearly every single pyridyl ketone reacts successfully. So we've only seen one or two so far that actually will not undergo this reaction. All right, so again, now that we have this, we want to extend that reactivity to do these other things that we demonstrated previously. It turns out none of these reactions work. The major product in all cases is that decarbonylation reaction. So that intramolecular reaction just outpaces anything that we can do to intercept these species. And so once again, this kind of begs the question, well, this could benefit from some mechanistic insight. And so thinking about this reaction mechanism, it turns out there's actually two potential pathways for this decarbonylation to occur. We could get rhodium coordination to the nitrogen and insertion into this carbon-carbon bond, which gives us this 5-membered rhodium metallocycle with the exocyclic acyl group. That then can undergo decarbonylation through a carbon monoxide migration and give us this rhodium species that undergoes reductive elimination. In contrast, we can also cleave that distal CC bond, the blue one over here, go through a 6-membered metallocycle, and then do the same carbonyl migration to give us this compound. So either one of these are possible. It's worth noting at this point that this is something that we didn't see for the quinoline, because through this same intermediate here, we get this 5-member metallocycle, but now the carbonyl is inside that 5-membered ring, and the decarbonylation process would lead to a 4-member metallocycle, which is unfavorable. So it just kind of shows why this is a different reaction pathway here. What we're doing to actually pursue this now is making a number of these different quinolynyl ketones with different substitutions on either ring, and then doing the mechanistic investigation, the kinetic investigation to see how they influence the reaction. So for example, on this blue side here, we'll put electron donating groups, electron withdrawing groups, and see what impact that has on the rate of the reaction. And do the same thing on the red ring side over here on the left side. And what we've seen is we've kind of gotten enough data at this point that we're really quite confident that the substituents that we put on the left side, so to speak, on the red ring, have a significantly larger impact on this rate of reaction. And so our most likely pathway is through this 5-member metallocycle. And now with that information in hand, we're going to go back and kind of apply that to see if we can develop and expand some of that reactivity that we showed earlier. And so shout out here to Grace, who this last summer really pushed this project ahead and has answered some of these questions for us. All right, moving forward, I just mentioned that order of activation and trying to figure out that project a little bit. We have started making some inroads and doing an exchange decarbonylation mechanism. So once again, we can start with a single pyridyl ketone and then take our choice of different aryl boronic acids, do that exchange and decarbonylation process. So once again, from one starting material can go to a whole number of different projects. And so shout out to Luke and Therese, who are currently pushing that project forward, and some, in addition to some other students who got that off the ground. And then also beyond that, we're also looking at further exploring directing groups. So that extension from quinoline to pyridine is a pretty small step, admittedly. And so we're now looking kind of at doing a systematic exploration of other groups and what things are possible to get these various reactions to happen. And so Claire and Charlie and Sarah have done some recent work in that area and are really and so at this point, I have to thank the students who have done this work. So in over 15 years now, or almost 15 years at Hope College, a lot of students have come through the doors of this lab. I will say that I have an open door policy here at Hope, is that if there's any student who wants a research experience during the academic year, if we can mesh our schedules to make that happen, I will make that work. And so what that does is it results in some students who come in and have a synthetic pathway and they get sucked in by the chemistry and become professional chemists. Others who come in, do a few things and slowly back away to something else, right? And I think that's really our job here at the PUIs, is not only just to train the next generation of chemists, but to really help the next generation of professionals figure out, where things are and where they're going. So the students here that are highlighted in blue are the ones who directly contributed to the projects that I've shown here. And thanks to all of them. I haven't done nearly enough to give shout outs to each of them as I've gone. So thanks to all of you. As I tell my students, I kind of steer the bus, but it's the students and their creativity and their enthusiasm that really drive it forward. And so thanks to all of them for all of their efforts. I also have to thank these groups for funding over the years. And once again, I'd like to thank you for the invitation today, and I would be happy to answer any questions.
Awesome. Jeff, great job. Thank you so much. And we already have a couple of questions lined up. So the first one I have here is from John Curto. He's one of my colleagues here at Pfizer. On slide 15, He was wondering if you could generate atrope isomers by having a 2,6 disubstitution of the aryl migrating in the decarboxylation.
Oh, yeah. So, yeah, I think we are definitely, we haven't seen a lot of that, but here definitely in some of these larger compounds with the aryl group, we get that decarbonylation, and we see evidence of two isomers that are have the same mass, but are definitely different from one another. So obviously, if these, we're assuming that they are essentially diastereomers, right? The two different atrope isomers. They're very challenging to separate, for obvious reasons. But yeah, we're definitely getting some of that action there. And that's one thing we're kind of playing with some ideas of if we could use a metal to bind them or something like that to kind of select one over the other. So lots of things on the drawing board, but yeah, that's a tough problem that I think is in front of us.
Excellent. Okay. And then a two-part question from Michael Doyle. The first one is, how difficult are the pyridine substrates to prepare, especially the orthoacyl analogs? And then second, which I think applies maybe to all of our speakers today, do students who work with you anticipate being an author when you have a publication?
Sure. So, yeah, and a shout out to Mike. Thanks for coming. Mike knows Hope College well and knows undergraduate research well. So thanks for coming, Mike. So what I would say is, first off, preparing these actually is really quite similar. What we've found is for most of them, we can do actually an orthoacylation. So a palladium catalyzed CH activation is what we use to make these species. So we can just simply start from the phenylpyridine and do an acylation reaction. Any of kind of what I would say the standard aromatic species, that works well for. It gets a little dicier. It's a couple of steps and a butylithium, lithium halogen exchange and so on to get to some of these heteroaromatic species. So those are a little bit more challenging to make. But any of these kind of straight aromatics over here are really pretty straightforward. Yeah, and in terms of authorship, that's one thing is with that open door policy, I make no promises at all about authorship in terms of students being co-authors on papers. I make it known that obviously if the students help push that project forward and things, you've seen that there's student co-authors on many of these different projects here. But I make no promises. And so we try to keep that separate from the experience. But yeah, there's obviously over the years, I've had a number of students who have contributed to these. And if they contribute a compound or two to these things, that's why some of these author lists are getting pretty long at times, is the students who contribute those final characterization data and things of that of these products are definitely included on the co-author list.
Excellent. I have a question actually about right here on this slide as well. I was wondering if you could if you used an amide, for instance, amides or other carbonyl derivatives instead of these benzamides, that could you get CN coupling as a product? So if you in what AR is, if you had a secondary amide, a tertiary amide, sorry.
Yes, at this point, so that's closest it would be to Michael Stoshak. I don't know is the pronunciation of that. At Rutgers has done some very similar things to that kind of decarbonylation. We actually have not tried it on this specific case. In other cases, we have not been able to do like ester or amide decarbonylation. But yeah, definitely something worth trying.
Okay, so I don't see any additional questions, and we are out sorry, one more. And what was your biggest challenge of developing a research program for PUI versus an R1?
I think probably any of us would agree, especially starting off, is just patience. Something that I could do as a postdoc in a week or something likely would take a student an entire academic semester, if not a year. So that's it. Is sometimes is just cranking down those timing expectations is really the main thing. I'm convinced that the undergraduate students can do anything technically and learn the procedures that a graduate student can, but it's just time.
Okay, and I would I'd like to say thank you so much to each one of you guys, to folks, to showing everybody what's going on at PUIs right now. The amazing research that you all are doing, I think is very inspirational. As part, being in pharma, one of the things we do is we go out and we recruit and we talk to students, and I noticed a few in the audience today of students that I've talked to personally over the last year or so who have indicated an interest in PUI careers. And I'm really excited for them to see all of the possibilities that you guys have shown them today, and that they can take that on and make that a career for themselves, and still do, and do some interesting research. And so I think it's been an inspiration for a lot of students that are watching today. And I know in a couple of cases, some former mentors of y'all's been watching, so I'm sure they're proud as well. So thank you again to Miles, to Rebecca, to Jeff, to Rio, the CCHF, and my colleagues at the ACS for giving me the freedom to put this session today together. I'm really excited that we had a ton of people watching, so thank you very much. And we'll be looking forward to seeing more from y'all soon, and whether here or at meetings and whatnot, hopefully in person sometime soon. So